Okay, so let's look at the steps of glycolysis and see what happens here. This is the first part or the first step of cellular respiration. We eat a bowl of cereal. That cereal is turned into glucose. A molecule of glucose is uptaken into one of our cells using the hormone insulin. That's why insulin is so important. Glucose cannot get into your cell without insulin. So you can have as much glucose as you want in your blood. You need insulin to get into your cell. When you get into your cell, you can then start to break it down. Okay, so when glucose gets into the cell, it's going to be broken down into two molecules of pyruvate, two molecules of a product called pyruvate. And we'll talk about how that happens, why we get two in a minute. But essentially, there are 10 enzyme catalyzed steps, so enzyme catalyzed reactions that happen in the cytosol or the cytoplasm of a cell to break glucose down to pyruvate. So this is like the first part of the journey here, where glucose starts to get broken down. And you can see how much effort is required already just in this part of the process. There are 10 steps to get glucose into pyruvate, and that's just a third of the process. Okay. Now, what we generate here are two net. I'm going to explain why that's net in a minute. We generate two net molecules of ATP. We also generate two molecules of NADH. Right? And this is what type of reaction is it if NAD plus goes to NADH? Is it oxidation or is it reduction? We gain some electrons. Is it oxidation or is it reduction? It's reduction. Perfect. Thanks. It's reduction. So if you go from NAD plus to NADH, that's reduction. Right? If you remember the mnemonic oil rig, so oxidation is loss, reduction is gain. I can help you sort of figure things out. We also, all right, so you generate two ATP and two NADH, and no oxygen is consumed. So this process does not require oxygen. That's going to be very important when we get to muscle metabolism. Glycolysis does not require oxygen, unlike the electron transport chain, which does. Okay, it's happening in the cytosol or the cytoplasm. And the net yield, the things that we make from glycolysis here, are those two pyruvates, two NADH plus those electrons, and then two ATP per molecule of glucose. Okay, now the NADH is going to be very important in shuttling those electrons. So you're going to see a common theme going on here. NADH is made in glycolysis, NADH is made in the Krebs cycle, NADH is made in that linker step. We have to make NADH, and all of that NADH is going to be shuttled off to the mitochondria to take part in oxidative phosphorylation. You can see the little arrow here, which is pointing out that these NADH that are generated here with these electrons are going to shuttle energy, those electrons have energy in them, to the electron transport chain to carry out oxidative phosphorylation. Okay, so the body is very efficient at doing that. It's going to use all of these steps to build up NADH, And when we study the electron transport chain, we're going to see why having NADH is so important. Alrighty? Any questions so far? Any questions? What questions do you have so far? Okay, so these are the general steps or the 10 steps of glycolysis. You're not going to be asked to memorize these steps just yet. If you're heading to medical school or nursing school or graduate school, you will have to memorize these steps. This is probably the most painful part of medical school. This and pharmacology, lots of memorization, memorizing all the steps, all the enzymes, all the cofactors, but you don't have to do that. What you have to understand is the general theme of what's happening for this class. Okay, we have glucose at the top, we have lots of enzymes catalyzing lots of different processes, and what we get at the end, for every glucose, we get two pyruvate. Right? We get two pyruvate. Now this process is broken down into two general segments, I guess. There's the energy investment phase, which is where we invest to ATP. And then there's the energy harvesting phase, which is where we harvest or create 2 ATP, 4 ATP, I'm sorry. So that gives us a net generation of 2 ATP. Because we had to invest 2 and we're making 4, we're really only using 2 net. I also want to point out here some of the names of some of the enzymes. So we talked about kinases last time. So for example, one of the very first enzymes here, hexokinase. Hexokinase has kinase in the name. What did we say kinases do?
Cleave.
No. The opposite. Come on, think about the opposite. Kinases, if it's not cleave, it's add, right? So kinases add a phosphate onto a molecule. Phosphatases cleave a phosphate from a molecule. Right? You want to remember that throughout this course. Kinases add a phosphate, phosphatases cleave a phosphate. Okay? So for example, we have glucose, hexokinase comes, it's going to attach a phosphate onto the 6th carbon of glucose, and that gives us glucose 6-phosphate, and so on and so on and so forth. All right, so another image of summarizing the steps. What I will point out here is the reason we have 2 pyruvate for every glucose is that this fourth step here, let's go back to this image. So this step, between fructose 1,6-bisphosphate, we generate glyceraldehyde 3-phosphate as well as DHAP, which is this molecule here. All right. Now, DHAP, dihydroxyacetone phosphate, is also converted to G3P. So as a result, we have G3P twice. So that's how we get that 2 from this point onward. We have 2 G3P because this molecule is also converted to G3P. So as a result, 1 glucose gives us 2 pyruvate at the end. Okay. Let's look at a question here. Let's look at this question. Let's see how we're doing. Okay. Okay, so we got majority saying B. You got B? Yeah, B, which is substrate level phosphorylation. Okay, so glycolysis generates ATP via substrate level phosphorylation. That is correct. And what did we compare that to again? What did we compare that to again? Stealing. Perfect. So taking the phosphate from something else. You can see that happening right here in this reaction, the steps of this reaction. All right, so there's a phosphate on this molecule, 1,3-bisphosphoglycerate. We're going to just take it right off and put it onto ADP and generate ATP that way. Generate ATP that way, sorry. Okay, now let's look at the linking step. So there's a small linker step between glycolysis and the Krebs cycle. And it's pretty short, pretty brief, but it's important. in turning that pyruvate that we generated into acetyl-CoA. Okay. So we have a pyruvate. So remember we generated 2 pyruvates. So we're going to multiply by 2 because this happens twice. But let's look at what happens to 1 pyruvate. Pyruvate is oxidized, or it's broken down, using the enzyme pyruvate dehydrogenase into acetyl-CoA, right? Into acetyl-CoA. So what happens is some of the carbons are removed. We generate carbon dioxide. We're also going to reduce NAD plus to NADH. We're going to put some electrons on that, and that is going to also go to the oxidative phosphorylation. So we talked about NADH already when we looked at glycolysis. We said that NADH is made or is reduced, and that has some electrons being stored on it. You can see that hydrogen there. And that's some stored energy that's then going to go to the mitochondria and take part in the electron transport chain where we have oxidative phosphorylation. So these processes are highly efficient in that this linker step does 2 things. It helps to generate some additional NADH. It also helps to convert pyruvate into acetyl-CoA. Now let's look at the Krebs cycle. The Krebs cycle. This is the last part of what we're going to talk about today. The Krebs cycle, also called the TCA cycle, also called the citric acid cycle. This is a cyclical pathway. Several enzyme catalyzed steps, several metabolic steps are taking place in this pathway. We start from oxaloacetate at the top, right? Acetyl-CoA is going to interact and become oxaloacetate. And then we're going to end right back up at oxaloacetate. That's why this is called a cycle. Okay? And what we generate here is some ATP. We also reduce some of the coenzymes that are involved in this process, which is adding some electrons to them. And then that acetyl-CoA is the initial substrate, and then what we get at the end is carbon dioxide, some more NADH, which heads to the electron transport chain, and FADH as well, and then some ATP. So I'll say that again. The whole purpose of the Krebs cycle is to take acetyl-CoA, to break it down, right, to make a cyclical process of metabolic reactions, to break it down. And what we really want to generate here is NADH, some more NADH, and FADH. We're going to see these 2 things in the electron transport chain. And then what we also get off are CoA, some ATP, and carbon dioxide. Okay? Carbon dioxide. Now, can you see where a lot of the carbon dioxide that we breathe out comes from? We bring oxygen in, and from all of these metabolic processes, we have to release carbon dioxide. So this is why we have to breathe it out, because every time a cell breaks down glucose to make some energy, it also generates some carbon dioxide. That's waste that we do not want. It has to go back into the blood, and it has to get to the lungs, and it has to be breathed out. Okay, so in our lungs, we exchange oxygen for carbon dioxide, and this is why. We're trying to get rid of it. It's a byproduct of most of the metabolic processes that take place in the